Hello everyone, welcome back to BioRenew Academy. Like, share, and subscribe my channel. This slide introduces cell culture media as the essential artificial environment needed to grow cells outside of a living organism in vitro. Think of it as the liquid life support system that provides everything a cell would normally get from its natural habitat. Here is how you can break down the main points, the three core pillars. The slide highlights three fundamental biological processes that the media must support: cell growth. It provides the building blocks for cells to increase in size and for populations to expand. Metabolism. It supplies the fuel for energy production and the necessary components for essential biochemical reactions. Division. It contains the specific signals and nutrients required for mitosis, cell replication. Why composition matters. The bottom section emphasizes that cell culture isn't one size fits all. Precision is key. The exact recipe of the media determines how healthy the cells are viability and how well they perform in experiments productivity. Sensitivity. Even tiny changes in the mixture can drastically change how the cells behave, which can lead to inconsistent experimental results. Key takeaway: the media isn't just cell food, it is a precisely engineered environment that dictates the success or failure of the entire experiment. Outlines of the seven major components of culture media, which are essential for sustaining cell growth in vitro. Here is a breakdown of how to explain each section. Core purpose. The slide categorizes the complex mixture of nutrients required to mimic a cell's natural environment into seven functional groups. Inorganic salts. These act as electrolytes to maintain osmotic balance, preventing cells from swelling or shrinking, and pH stability through buffering. Carbohydrates, typically glucose, these serve as the primary energy source for cellular respiration and metabolism. Amino acids, the fundamental building blocks for protein synthesis. Since cells often cannot synthesize all of them, they must be provided in the media. Vitamins, these function as cofactors for various enzymatic reactions, ensuring metabolic pathways run efficiently. Lipids, necessary for cell membrane formation and long term energy storage. Proteins and peptides. This group includes growth factors that signal cells to divide and attachment promoters that help cells adhere to the culture vessel. Trace elements, micronutrients like zinc or copper required in tiny amounts for specialized cellular functions and enzyme stability. Inorganic salts are the role of inorganic salts in cell culture media, describing them as the ionic framework necessary for maintaining a stable environment for cells. Core functions are: inorganic salts perform four vital regulatory roles. Osmotic balance. They regulate the movement of water across cell membranes to prevent cells from shrinking or bursting. Membrane permeability. They control how substances enter and exit the cell. pH buffering. They stabilize the acidity by alkalinity of the media, which is crucial for cell survival. Enzymatic activity. They provide the necessary ions that act as keys to turn on various enzymes. Key salt examples. The slide highlights four specific salts and their primary biological purposes. Salt primary role: sodium chloride NaCl, the main electrolyte used to maintain osmotic pressure and the electrochemical gradient. Potassium chloride KCl, essential for maintaining the membrane potential electrical charge and activating enzymes. Calcium chloride CaCl, too crucial for cell to cell adhesion, sticking together, signaling and overall structural integrity. Magnesium sulfate MgSO, 4A, necessary cofactor for enzymes involved in ATP energy metabolism. Carbohydrates, energy currency of cells. Carbohydrates are the primary energy source for cells, fueling metabolic processes through cellular respiration. In cell culture systems, glucose is the most commonly utilized carbohydrate, primary role fueling metabolism. Carbohydrates are broken down to produce ATP, the energy currency that powers cellular activities. Cellular respiration, glucose is oxidized through glycolysis and the citric acid cycle to release stored energy. 5 per glucose concentrations in media. The concentration of glucose is critical for maintaining optimal culture conditions. Media type concentration: primary use high glucose media 4.5 gram per liter, used for rapidly dividing cells. Low glucose media 1 gram per liter, used for specialized applications. Critical consideration: excessive glucose leads to lactate accumulation and pH drop, whilst insufficient glucose restricts cell growth and product. Witty balancing glucose levels. are essential for optimal culture conditions. Amino acids, protein building blocks. Amino acids are fundamental components of cell culture media, serving as the primary building blocks for proteins. Classification: essential amino acids. These cannot be synthesized by the cells and must be supplied externally in the medium. Non essential amino acids. These can be produced by the cells themselves through various metabolic pathways. Critical examples: L-glutamine, often considered the most important amino acid in culture, though it is famously unstable in solution. L-lysine, an essential amino acid crucial for maintaining protein structure. L-methionine, a sulfur-containing essential amino acid. Important note: because L-glutamine degrades into ammonia, which can be toxic to cells, many laboratories opt for stable dipeptide alternatives such as L-alanine L-glutamine. Vitamins, enzymatic cofactors. Vitamins are essential nutrients that cells generally cannot produce on their own in sufficient quantities. Core function: they primarily act as cofactors, helper molecules in enzymatic reactions. These reactions are vital for maintaining cellular metabolism and ensuring overall cell survival. Requirement: while only needed in minute quantities, their presence is non-negotiable for complete media formulations. 4. Key vitamin groups highlighted: vitamin B complex includes B1 thiamine, B2 riboflavin, B6 pyridoxine, and B12 cobalamin. Role: supports energy metabolism and the synthesis of nucleic acids DNA by RNA. Vitamin C ascorbic acid. Role: acts as an antioxidant to protect cells and serves as a cofactor for collagen synthesis and iron absorption. Consequences of deficiency: without adequate vitamins, cell cultures will experience reduced growth rates, metabolic dysfunction, eventual cell death. Lipids, membrane architects. Lipids are essential components in cell culture media, acting as a structural foundation and functional regulators of cellular life. Primary function: cell membrane formation. Phospholipids and cholesterol spontaneously assemble into a lipid bilayer, providing the physical boundary and structural integrity for the cell and its organelles. Energy storage. Triglycerides serve as a concentrated, high energy reservoir that cells can mobilize for long term growth and survival. Signal transduction. Many lipid derived molecules function as messengers in complex cellular communication pathways, regulating processes like growth and inflammation. Key components: fatty acids exist in saturated and unsaturated forms to maintain optimal membrane fluidity. Cholesterol, a vital sterol that modulates membrane rigidity and facilitates the formation of lipid rafts for specialized signaling. Phospholipids, the primary structural building blocks that form the amphipathic water-loving by water-fearing framework of the bilayer. Noted: lipids are especially critical in serum-free media formulations. Because they are not naturally provided by serum, they must be explicitly supplemented to ensure healthy cell development. Proteins and peptides. Growth promoters, proteins, and peptides are essential components in cell culture media, acting as key regulators for growth, health, and stability. They serve four primary functions: cell growth promotion. Growth factors like EGF, FGF, and IGF are signaling proteins that stimulate pathways for cell proliferation and differentiation. Cell attachment. Adhesive proteins such as fibronectin or RGD peptides facilitate the binding of cells to culture surfaces, which is critical for anchorage dependent cells. Nutrient transport. Carrier proteins bind to and deliver essential molecules like iron or fatty acids that cells cannot easily absorb on their own. Protection. These molecules help shield cells from mechanical shear stress, especially in dynamic environments like bioreactors. Key protein types: component function. Albumin, the most abundant serum protein. It binds fatty acids by hormones and reduces surface tension. Growth factors, for example, EGF, FGF, IGF specifically stimulate growth and proliferation pathways. Hormones, for example, insulin, transferrin regulate metabolism and essential nutrient transport like iron. Trace elements. The final essential components, trace elements, are micronutrients required in extremely small quantities but are absolutely essential for specific cellular functions. 4. These include metals such as iron, zinc, copper, manganese, selenium, and molybdenum. Key roles: iron, oxygen transport and electron transfer; zinc, enzyme cofactor and transcription factor; copper, oxidative enzymes and cross-linking; selenium, antioxidant enzyme activity. Media formulation summary. This table summarizes the primary categories of components needed for cell culture media and their respective functions. Component primary function: inorganic salts, osmotic balance, and pH. Vitamins, enzyme cofactors, trace elements, specialized functions, carbohydrates, energy source, amino acids, protein building, lipids, membranes, proteins, growth factors. Thank you for your support. Please comment on my video and get new topic.